The overall goal of this procedure is to determine the concentrations of nucleic acids and proteins accurately and reproducibly using a microvolume spectrophotometer. This is accomplished by first selecting the appropriate path length for the sample to be measured. The second step of the procedure is to measure a blank. Next, the DNA or protein sample is measured. The final step of the procedure is to save the data in PDF or CSV format. Ultimately, highly reproducible results for DNA and protein quantitation can be obtained rapidly and accurately by this method.
The main advantage of this technique is it's fast, precise, and reproducible.
To begin the procedure for DNA quantitation, turn on the BioSpec-nano Spectrophotometer. Open the BioSpec-nano software from the PC desktop. Click on the Simple Nucleic Acid dsDNA tab. The software will establish communication with the instrument and run an initialization sequence. This verifies that the instrument is performing according to specifications. Next, select the path length on the path length slider. Three options are available. For microliter volume samples, 0.2 mm or 0.7 mm can be used, and for dilute samples, there is an option to use a 5 mm path length cuvette. In this demonstration, 0.7 mm is selected for measuring a 2 microliter sample. All calculations are based on the path length selected. Next in the software, click the Setup tab and select Auto Wiping Setup. Select one for one wipe.